Hello and welcome to Genomics Workshop number 1. My name is Dr. Andrew Thomas. I'm a Principal Lecturer in Biomedical Science at Cardiff Metropolitan University. So in this workshop we're going to be talking about the human genome. We're going to be talking about some of the genes in the human genome, and we're going to access the database to look at some DNA sequences. So I'm going to start by asking you a question, and the question is this: what major scientific event happened in April 2003? Well, the clue is in the title of the workshop, and you probably guessed by now. It's the completion of the Human Genome Mapping Project, and this took place in April 2003. It took the whole of 13 years to complete. Started in 1990. Finished in 2003, and there was hundreds of thousands of scientists involved in sequencing the human genome in countries all over the world, and it cost an approximate $300 million to complete. But the good news is we can sequence a genome much quicker than that now. We can do it in about 2 days, and it only costs about £1,000. So what does the human genome look like? Well, we used to think there was hundreds of thousands of genes, but actually there's only 21,000 genes that are needed to make a human, and they're arranged on our 23 chromosomes. But the amazing fact is they only compose 2% of our genome. The other 98% of our genome is what we call junk DNA or dark DNA, and we don't fully understand what the function of that DNA is. So if we look at a pie chart representation of the human genome, you can see that our genes just represent this thin yellow slither here, and then the rest of the genome is composed of other sequences that are not genes. The green sector here is actually viral sequences, and these are remnants of old viral infections that Homo sapiens have had, tens of hundreds of thousands of years ago. So what do genes do? Well, we all know that genes code for proteins. They're short pieces of DNA coding for proteins, and they also code for what we call functional RNA molecules. So this diagram underneath is what we call the central dogma of molecular biology. So the double helix here represents the gene, and a gene is transcribed into messenger RNA. So the messenger RNA carries the DNA code into the cytoplasm of the cell, and once it's in the cytoplasm of the cell, it's translated into a protein, and the organelles in the cytoplasm that read the messenger RNA are called the ribosomes, and they read 3 bases at a time, and they translate those 3 bases into amino acid. So this diagram here represents the amino acids that are arranged on a polypeptide chain, and these polypeptide chains fold up into a mature protein, and that mature protein can be an enzyme or it could be a structural protein. So what do the human genes look like if we focus in on them, zoom in on the molecular structure? So every gene starts with what we call a promoter region, which is like the regulatory region or the on-off switch, and then we've got an alternating set of sequences called exons and introns, and they repeat all the way down the length of the gene until the gene stops. And every gene has got a start and it's got a stop. And the interesting thing is the introns are part of the non-coding 98% of our genome, so they have to be spliced out. So we've got enzymes that cut out the introns, which are the red sequences in this case, and leave us with this messenger RNA molecule here. And then the RNA molecule is what gets translated into the protein. So how are the genes arranged on chromosomes? So if we take a chromosome, we unravel it like a ball of wool, and we look at the length of DNA double helix, we see that the genes are arranged sporadically throughout the chromosome, and then there's huge areas of non-coding DNA between the genes. So if you look at a diagram of chromosome 10, for example, which is 135 million bases in length, there's a whole list there of all the genes that have been identified on chromosome 10. and we have approximately about 1,000 genes per chromosome. So can you give me the name of a gene? Can you think of a gene that springs to mind? Well, I'm going to give you 6 examples here that I've just chosen randomly. I could have chosen any one of the 21,000, but I thought I'd try and choose some that you know and some maybe that you don't know. So the first one I've got there is insulin, which you know is a peptide hormone produced by the pancreas, and that is responsible for signaling to muscle cells to take glucose up from the blood. Keratin is a gene which makes a protein called keratin, which is part of our hair structure and our nails. Tyrosinase is an enzyme found in our skin which makes melanin in our melanocytes, so it allows our skin to go brown in the sunlight. Amylase is an enzyme found in our digestive juices, in our saliva, in our intestines, and it breaks down starch into glucose. The last 2 genes, BRCA1 and P53, are classed as tumor suppressor genes, and these are unusual genes in that they help our cells to avoid cancer. So they're really important in terms of the protein, because obviously they prevent our cells becoming malignant. And if you get a defect in one of these genes, so BRCA1 will predispose you to things like breast cancer if you get a defect in that particular gene, and P53 is a protein that is in all our cells, and it protects all our cells from becoming malignant. So it's important that we don't get mistakes in these genes, and we're going to look at P53 in a bit more detail in a minute. So how can we look at genes and the proteins they code in more detail? Well, the human genome has been mapped, and that information is freely available to anybody in the world to go and have a look at. And what we're going to do today is we're going to explore the human genome using a database called the National Centre for Biotechnological Information, or the NCBI database. And this is the URL, and we're going to go there now. So if we upload Google and we type in NCBI into the search box, it brings us up the hit for the NCBI database. And this is the home page. It's not very exciting, but behind there we have the human genome at our fingertips. So there's a little button up here that says all databases, and if we click on that, it gives us the list of the databases that are available. And one of the databases is called Gene. So we're going to click on Gene, and we're going to type in the gene we're interested in. And I'm interested in the gene P53, and it's a human gene, so I'm telling it to go look in the human genome database for this gene. And then it pulls up the entry for tumor protein 53. So I'm clicking on this, and I'm waiting for it to bring up the entry now for P53. Just going to hide this sidebar. So here's some information about the P53 gene. It's called a tumor protein, and its full name is tumor suppressor protein, and it's involved in protecting us against cancer. And if you get mutations in this gene, it can actually give you cancer, because obviously it protects us against cancer. So if it goes wrong, then that's not a good thing. It's located in chromosome 17, and we know it's got 12 exons. And if we come down further, we get a diagram of chromosome 17, and the red line represents the gene, the red arrow, and then further down we get a bigger diagram with a blue line, and the blue line is the chromosome, and the green lines that we see here are the RNA molecules that are derived from that gene. So this is the start of the gene over here, and this is the end of the gene over here. And you notice there's lots of different RNA molecules, and that's because every gene in the human genome is capable of producing different proteins or different types of proteins from the same sequence. But we'll leave that for another workshop. So this is the P53 gene, and if I click on it, I can move it from left to right. What I'm going to do is just put it into the middle of the screen. And I'm going to do that because I want us to zoom in on this start of the gene here. So I'm going to go now to a button here called Tools. I'm going to click on Tools, and I'm going to come down to a button which says Sequence Text View. And I'm going to click on that, and it's going to bring up the DNA sequence. And I'm going to make that window bigger. And what you notice about the DNA sequence is that it's coloured in different colours. You've got an unhighlighted region here, and this is the extragenic DNA or the junk DNA. And then as soon as it gets coloured, this is the start of the gene. So this is the start of the P53 gene. And if I go to the next page, by clicking next page, what I find is that you get a green section, and the green section represents what we call the introns. And then the blue section is the start of the exon. And then when the exon starts coding for amino acids, it goes a kind of pink colour, orangey pink. And what you notice is that the first amino acid that is read in a gene is always the same amino acid. In this case, it's methionine. So every gene in the human genome will start with the letters ATG, and ATG is the codon for methionine. And then you've got the first exon, followed by an intron, followed by an exon, followed by an intron, followed by an exon, followed by an intron, all the way through the gene until you come to the end of the gene. So this is the last exon, and you'll see the last exon finishes with a star, and that star represents the stop codon. So the ribosome is given the start codon being ATG, and in this case it's given a stop codon TGA. And there's 3 different types of stop codons that is available to the ribosome to know when to stop translating the RNA into protein. So that's the gene and the structure of the gene. What I want to do now is I want to take you to have a look at the protein. So I'm going to go to the protein database, and I'm going to go click on the word protein, and I'm going to type in P53 human. And this is going to take us to the protein database now, as opposed to the DNA database. And here's the entry for P53. And slightly different information, because instead of DNA sequences now, what you'll notice is we've got an amino acid sequence at the bottom here. And you can see it starts with methionine, and then these are the different amino acids. And it's about 400 amino acids in length. To the right here, you've got a nice pretty diagram, which is a graphical representation or a computer generated representation of what the protein looks like. If you were to zoom in with a really big, powerful microscope and look at the protein. Now, as I said, this protein is a protein which protects us against cancer. And we'll look at it on the full screen because it's quite interesting. And what you can do is you can move the protein around. You could look inside it, and you could look on the side of it, and you could look on the other side of it. So the computer has generated this 3D model from looking at the different amino acids in the actual protein. So some amino acids will form what we call alpha helixes, and some amino acids will form something called beta sheets. And these 3-dimensional shapes are important in the function of that protein in our bodies. And if we get mutations in our DNA, and those mutations cause those amino acids to be replaced by other amino acids, then we can have defective proteins, and defective proteins could result in some sort of pathology like a cancer. So these proteins are really important in keeping us healthy. But we'll talk about mutations and other things in another workshop. Thank you for listening, and I hope you enjoyed it. Bye for now.
Hi everyone, my name's Georgie. I'm a second year student studying biomedical sciences at Oxford University. Just a little bit about me before we go any further. I'm from Cardiff. I went to Ysgol Gyfun Gymraeg Llantab from year 7 all the way up to year 13. My A-levels, I did biology, chemistry, psychology and Welsh Bac, and to AS I did French. So when I was in year 12, I wasn't really sure what I wanted to do at uni because I'd always really loved languages, but I also really enjoyed science, which is kind of polar opposites. But when I was doing my research, I was looking around at different courses and then I found neuroscience, and it just seemed like the perfect fit because I've always loved biology, loved chemistry. I did psychology A level, as I said, because we didn't do it at GCSE, and I thought that was fascinating. I really enjoyed that. And neuroscience just seemed perfect because it seemed like a perfect mix of all three. And also, I've always been someone who's really enjoyed processes and mechanisms and sort of fitting the little bits of the puzzle together to make the whole picture, which really is what neuroscience is because it's such an up and coming field and there's so many new developments all the time. So I decided on neuroscience. As I said, I was looking at my different courses, looking at my different options. Once I started going to SED events and I realised that, Oxford was an option for me, I realised sadly that Oxford doesn't have a straight neuroscience undergraduate degree. It's got a postgraduate degree. But they do have biomedical sciences, hence why I'm here. So with Biomed at Oxford, your first year is very broad, but after that you pick certain modules and you can come out with a neuroscience degree. So as I said, Biomed at Oxford is a really, really broad course, which was one of the things that drew me to it the most in the first place, and it's one of the things that I'm enjoying the most about it now. So on average, the cohort per year is around 40 students, which is quite small for an Oxford science course. Biology, for example, they have around, I think, about 100 students per year. But it's really nice because you get to know everyone really well, and it's a really good way to make friends outside sort of your immediate friendship circle or outside your college. So yeah, first year you do a lot of different modules. The core ones, I believe, are brain and behaviour, genes and molecules, body and cells, and statistics as well, which sounds intimidating. At least it did for me because I didn't do A-level maths, but it's really not that bad. The lecturers are really, really good and you get lots of help. So it's definitely manageable. You don't have to have maths to be able to get through that. Some general examples of things that you get covered in those modules. You do the visual system in the brain, you do the lungs, the kidneys, the heart, the digestive system, you do DNA replication, transcription, translation, gene expression, any other ones that I enjoyed. The auditory system, that was really cool, and some general anatomy as well. You have anatomy classes in the demonstration room of people who've kindly donated their bodies to scientific research. So you get to really sort of see all the anatomy and the processes that you're learning about in real life, which is really, really cool. It's a really good opportunity. So yeah, first year is quite busy. I'd say an average week in the life, I had about 3 to 4 lectures a day. They tend to be in the morning, which is kind of hard to start with, but you get into a routine of waking up for 9 a.m.s. So yeah, 3 to 4 lectures a day, Monday to Friday. Once a week, we would have a 2 to 4 hour practical in the afternoon, which was relating again to the topics that you'd cover in your lectures. I also had anywhere between 1 and 3 tutorials a week. So this is, I guess it's an Oxford unique thing. I think other universities have a similar sort of system where you get set an essay or a problem sheet full of questions about a week in advance. You work through them by yourself. Obviously, you're more than welcome to email your tutor to ask questions if you need, but you work through them, you do your research, you write your essay, you submit it, and then you go to the tutorial and you just kind of discuss the topic in general, discuss what you wrote, what you found interesting. It's an opportunity to ask any other questions that you had from your reading that you couldn't find an answer to. So I know they seem quite intimidating to people to start with, and I was really scared before my first tutorial. But they're a really good opportunity, and it's really fascinating because you have a chance to talk to someone who's the best of the best in their field, and try and sort of absorb some of their knowledge. So it's really fun. So yeah, first year is really broad. At the end of first year, you're supposed to have these exams called preliminary examinations, or everyone calls them prelims. Fortunately or unfortunately, it depends on which way you look at it. Mine got cancelled because of the pandemic. But those exams consist of three, I believe, 2.5 hour papers. Don't quote me on that, but yeah, three papers on where you write essays for body and cells, genes and molecules, brain and behavior, two each. You also are supposed to have a 3-hour statistics exam, I believe, where you have a choice of lots of different questions and you choose which ones you want to do. And there's also a multiple choice exam as well. So second year is when you get to start to specialise, as I mentioned earlier, to confirm whether you're going to go for a neuroscience degree or a self-assistance biology degree. So you get a choice of 20 modules, I think, in the summer after the end of first year, which is an amazing amount of choice. And that was another thing that I really, really loved, and that really, really drew me to the course. So some examples of things that you can cover. You can do immunology and microbiology, cellular pathology, so things like atherosclerosis, HIV, etc., etc. You can do hormones, endocrinology, auditory neuroscience, visual neuroscience, sleep, neuropharmacology, protein structure. Yeah, loads and loads of different choices, which is really, really good. This year, on average, I've had about 1 to 2 essays a week and 6 to 10 lectures a week, which again is slightly bit less busy than first year, but the essays are a bit longer, require a bit more research, a bit more thought put into them. You have part one examinations at the start of the summer term of second year. Then you start your research project, which carries into third year. Third year, you finish up your research project and do some extra modules to confirm your degree choice, and then do your final exams, and then that's you done. So careers-wise, I have to admit I'm not entirely sure what I want to do after my degree yet. But the uni has a really good careers service that sends you lots of emails, and they're always on call to have an appointment and have a chat and sort of talk you through your options. With the biomedicals, I know a lot of people tend to go into research because you cover so many different topics. You have so many different options of the different pathways you can go down, research or otherwise. So yeah, research is the primary option. I know a lot of people also go into postgrad medicine. Because we do in first year share a lot of our lectures with the medics, so we kind of have a similar foundation. Or of course, there's a master's or a PhD, or everything and everything in between. There really is no sort of one set career path coming out of biomed. So, as I hope I've been able to convey by now, I've been really, really enjoying my time here at Oxford. I love the variety of the course, how unique the topics are, all the different things, and how much detail you go into. The fact that you can essentially tailor the degree specifically to what you're interested in, I think is incredible. Oxford in general is just such a beautiful city. The collegiate system that the uni has really makes your college feel like home, and it gives you a really good support system of such a tight-knit community of such lovely people. So yeah, I've really, really been enjoying it. I would say my biggest advice in general for uni would just be to throw yourself into it. I know this is quite cheesy, but there is no such thing as a stupid question. Ask the questions that you need to ask. Chances are someone else in your seminar or your lecture hall or your tutorial was wondering the exact same thing. So yeah, I would say you only get out of it what you put in. Secondly, everyone's in it together. Your course mates aren't your competition. Work with them, share the workload. It makes everyone's life so much easier. But I also think most importantly, make sure that you have things to do outside of your degree. Your work isn't the be all and end all. It's not everything that you're at uni for. You are at uni, for the experience, and you need to look after yourself. So I think trying your very best to do things outside of your college, outside of your accommodation, meeting new people, having that healthy balance, I would say, is one of the most important things. So yeah, that's all from me for the time being. Thank you so much, all of you, for listening. I hope what I've been saying has been helpful and has made some sense. I hope I haven't waffled on for too long. So yeah, I'll now hand you back to the host who will tell you about your next steps. Thank you very much.
Gwerthfawr tu hwnt, dwi'n siŵr. What an insight those masterclasses were. Ac mae'n werth I chi fyfyrio arnyn nhw. You now have 15 minutes to reflect on both masterclasses. Time to think about which subject you preferred and why. Can you see yourself studying either of those disciplines at university? Wnaethon nhw gadarnhau mai dyna'r pwnc I chi? Remember that realising something is not for you is just as valuable as finding something new to consider. As you work through the reflection template, think about what you've learned, what you liked or maybe didn't like, and how what you've seen has changed your thinking about what course you want to study. Cwestiynau mawr, ond peidiwch â phoeni, please don't worry. You don't need to have all the answers now, but reflecting like this will help you start to decide what subjects are, and importantly, aren't right for you. Ac fel y soniais I, as I said, if you don't have the resources already, you can download these from the lobby to complete. Welai chi nôl fan hyn wedyn. I'll see you back here at 3:40.